and that is about the DNA replication in case of prokaryotes. Can you see the slides on your screen?
Yes, sir.
Okay. In previous lectures, we have discussed about the replication machinery and all other accessories which are involved in process of replication, and we have also discussed about various enzyme and enzymatic machinery and proteins which are performing their role during the process of replication. So if we talk about the replication process in case of prokaryotes, there are three stages of replication. And these three stages are initiation, elongation, as well as the termination process. So if we look at all these processes one by one, initiation, it basically occurs at the origin of replication. As all of you know that replication always start at specific point on the double-stranded DNA, where the two strands of the DNA, they are separated from each other and they form a fork which is called a replication fork, and it can also be called as origin of replication, and that specific site will be called as ORIC site. So in the process of during the process of initiation, this process occurs at the ORI site or origin of replication site, and this will separate the double-stranded DNA. Once the double-stranded DNA is separated from each other at a specific point, a fork-like structure will be formed, and in the next step during the process of initiation, primer will be synthesized. As all of you know that primase enzyme is involved in the synthesis of primer. Primer is basically a short ribonucleotide structure which could be made up of 10 to 12 ribonucleotide. This is basically RNA in nature, and it is required to initiate the process of replication. So the process of replication can't be initiated in the absence of primer. Once the primer has been synthesized, the replication fork has been formed, and the DNA polymerase along with all other enzymatic machinery that has been deployed to the origin of replication site. Now this replication process will enter into a next phase of the replication, and that phase will be called as elongation. So elongation, it involves the addition of new nucleotides, DNTPs, and the new nucleotides will always be added on the basis of the complementary of the template strand. Complementarity mean the new base will be added according to the complementary bases which are present on the single-stranded DNA. So in the elongation phase, new nucleotide will be added, and once the nucleotide will be added to the third prime end, it will form the phosphodiester bond with the 5 prime end of this nucleotide and third prime end of the already present nucleotide, and say phosphodiester bond will be formed, and ultimately the DNA strand will be extended. So this is the next phase of the replication, which will be called as elongation. And last phase of the replication is termination. In case of termination, the replication process will be stopped at specific point, and those specific points are called as specific termination sites. Those will be called as replication termination sites. So those sites, when those sites will reach, the process of replication will automatically be terminated. So these are the three phases of the replication. Here, this is genome of E.Coli. Here you can observe that there are various genes which are present. This DNA C and variety of the other genes, they are present. So this is ORI site, origin of replication. This is the area from where the process of replication will start. And as all of you know that E. coli is a prokaryote, it is a bacteria. So prokaryotic DNA is mostly of the circular nature. So that is the DNA of the E. coli. And here is the site which is called ORI site. This is origin of replication, from where the process of replication will be initiated. So if we look at all these phases into detail, first in case of initiation, as I have told you that replication will always start at a specific point, and that specific point will be called as origin of replication or ORI site. And structure of the origin is basically this is the ORI site. This is 248 base pair long structure, and this is basically AT rich. It is basically rich in A adenosine and thymine. Adenine, thymine, they are most frequently present in this site. So one is called 13 MER sequence. This is 13 MER sequence, uh, and they are tandem array of the base pair sequence. Tandem array mean that these base pairs or these bases, they are repeated. This is called tandem array. I mean head of one is when it joins with the tail of the other. This is called tandem repetition. So this is tandem array of the repeats, that these nucleotides, they are repeated uh, regularly, and this whole structure is consisting of 13 nucleotide sequence. This is one consensus sequence. These are called consensus sequences. Why we are calling them as consensus sequences? Because they are more or less present with same arrangement in almost all of the prokaryotic cell. That's why those are called consensus sequences. So one consensus sequences are 13 MER, and this is 9 MER sequences, and they are again having they are having the binding site for DNA A protein for 9 base pair sequences. Such type of the 4 base pair sequences, they are present regularly at different sites on the DNA, and they are actually having the binding site for DNA A. As all of you know that DNA A is basically the helicase enzyme. Once these sequences are present there, the helicase enzyme will bind there, and it will start the unwinding of the double-stranded DNA. So these are the so it means that in case of prokaryotes, the process of replication will always start at specific sequences on the DNA. Those sequences are called as consensus sequences, and there are two types of the consensus sequences. One are 13 MER sequences, which are and both of them are AT rich region. Basically 13 consensus sequences, they are consisting of 13 base pair, and 3 such type of the sequences, they are present in different region. And if we look at the 9 MER sequences, there are around about 4 such type of the structure which are present at regular interval on the DNA, and these 9 MER sequences, they actually represent the binding site for the DNA A, which is basically the helicase enzyme. It will bind and it will start the process of unwinding of the double-stranded DNA. So this is another structure. This is it will be called as a preprimosome. This is formation of the preprimosome. As here you can see, this is DNA A. DNA A will bind to this sequence, which is called 9 MER sequence, and a complex will be formed, and with the help of the ATP, the complex will be opened, and once another ATP molecule is utilized. Here you can see this is DNA C helicase enzyme, and this is DNA B. This is helicase enzyme, and this is DNA C, another type of the enzyme. So with the utilization of the ATP, now DNA B will bind. B will start the unwinding of the double-stranded DNA. So the helicase is basically performed by this DNA B, not DNA C. So DNA B, in the first step, DNA A will bind, and in the later stages, this ATP utilization will result in the binding of DNA C and DNA B. And DNA B is actually helicase enzyme. When DNA B will bind, DNA C will automatically be removed from there with the utilization of the energy, and a structure will be formed which is called pre-priming complex, mean this structure is formed before the synthesis of primer. That's why it is called pre-primosome, mean the formation of the complex which is formed before the so once the double stranded, as all of you know that once this double stranded DNA that is unwinded, the two strand they are separated from each other. Next step is the synthesis of the primer. So because this structure is actually formed before the formation of the primer, that's why it will be called as pre-primosome structure. This structure will be called as pre-primosome structure because of the nature of the formation of this structure. So now you must have a clear idea about these consensus sequences, that there is 13 MER sequence and there is 9 MER sequence, and there are 4 such type of the sequences are present on different regions of the DNA, and similarly there are 3 such type of the 13 MER sequences, they are present on different sides of the DNA. And as we have discussed in the previous slide, that these MER 9 MER sequences are consensus sequences. They are having the binding site for DNA A. Once the DNA A will bind, it will stimulate for of binding of certain other DNA C and DNA B, and DNA B is helicase enzyme. So in the with the utilization of the energy in the form of the ATP, this DNA B will start the unwinding of the DNA double-stranded DNA, and this structure will be formed before the formation of the primer. That's why it will be called as pre-primosome structure. So if you look at the origin of replication, as here you can see that this is parental DNA strand, there are 2 DNA strand, double stranded DNA, and this is the site where the process of replication will start. This will be called a origin of replication, and here is replication complex will be formed. Complex mean this will having the primer and all the replication machinery, all the enzymes which are required for the process of replication, all of them they will bind to this specific position which is called ORI site, and they will form a complex which is called replication complex. So at this specific region, a initiator protein will bind, and this initiator protein it will start the unwinding of the DNA. So this is the site actually on the double-stranded DNA where the replication process is started with the help of certain enzymes and proteins which are involved in the process of replication. So if we look at the replication fork formation, as we have discussed in previous slide, that DNA A actually recognize the ORI site or origin of replication, and DNA B or helicase and DNA C, they basically join the DNA complex, DNA and DNA A complex, and open the AT rich region and move on template downstream further to separate enough space. So that's how basically the initial molecule or initial enzyme which bind with the double-stranded DNA is DNA A. This DNA A is involved in the recognition of the origin of replication site. When ORI site is recognized by this DNA A, this DNA A, once it has recognized the ORI site, it will stimulate the DNA B, which is helicase, and DNA C. So DNA C will stimulate, or it will facilitate the binding of the DNA B to the ORI site, and DNA A is always already bound there. Here you can see that this is DNA A is always there. DNA C is actually facilitating the binding of this DNA B to this DNA and DNA A complex. This is DNA A and this is DNA. So DNA and DNA A complex, it will be joined by this DNA B with the help of DNA C. DNA C will facilitate the binding of this DNA B to the DNA A and DNA complex, and once this DNA B, which is basically helicase in nature, once it binds with this double-stranded DNA, it will start separating the 2 strands from each other, and hence the replication process will start there. So DNA A is gradually it is replaced, and now once enough of the 2 strands they are formed as a result of the helicase activity of this DNA B. Now in the next step, as all of you know, that single-stranded binding protein SSB. Now SSB will bind to the single-stranded DNA to provide the stability of the DNA as long as it is present in single-stranded form. So next step is, after the helicase activity of the DNA B, next step which will be included in the initiation phase is the primer synthesis. As all of you know that primase, DNA primase or RNA primase is an enzyme. It will join and it will form a complex which is called primosome. So the initial complex was pre-primosome, and after the pre-primosome complex has been formed, the two strands they have been separated from each other. Now the next phase or the next stage is the formation of the primosome. Mean RNA primase or the DNA primase will synthesize the primer, and primer will bind to the separated DNA strand, to the single-stranded DNA, and this structure will be formed as primosome. So primase start the synthesis of primer on the single-stranded DNA template, and it will utilize the NTP nucleotide triphosphate as the substrate in the 5' to 3' direction, and this primase will also require the energy in the form of the ATP. So short RNA fragment, it will be synthesized, which will be called as primer, and this primer will also have the 3' hydroxyl group. Mean it will also have, just like that of the DNA strand, the RNA strand or the primer will also have the free hydroxyl group at its 3' end, and at this 3 hydroxyl group, new incoming nucleotides they will be added, and hence the DNA strand will be elongated. So this is another diagram of the initiation process. Here you can see that we have a leading strand as well as we have a lagging strand. So in case of once the replication fork has been formed, origin ORI site has been identified by the various enzyme, and helicase activity was performed by the DNA B. Now we have two strand from 5' to 3'. The strand which is running in 5' to 3' direction. Here the synthesis of new DNA strand will be in continuous fashion, and that strand will be called as leading strand. This is short fragment is primer. This is synthesized by the primase enzyme, and by using the free hydroxyl group or free 3' end of this primer, now new nucleotide or deoxyribonucleotide DNTPs will be added, and overall this strand will be elongated. If you look at the other strand, this is the and here you can see the single-stranded binding protein. They are also there. If we look at this strand, this is actually running in 3' to 5' direction, and as all of you know that DNA synthesis will always be from 5 to 3' direction, because new nucleotide or new DNTP can always be added, can only be added at the 3' end. It can't be added to the 5' end. That's why on this strand, the primer from the primer, the short fragments will be synthesized, and those short fragments will be called as Okazaki fragments, and the synthesis of the DNA on the lagging strand will be in discontinuous fashion, and that's why it will be called as lagging strand DNA. And later on, these gaps they will be filled by the DNA ligase enzyme. So that's how there is difference between the leading strand DNA synthesis and lagging strand DNA synthesis. Once the initiation has started and primer has been synthesized and new DNTPs they have been added to the growing DNA strand, now DNTPs are continuously connected to the primer and or the nascent DNA chain by DNA polymerase 3. Here you can see that these are the various structure. This is growing strand. Here it is having 3' free end. Now new DNTP will be added here, and hence it will be elongated. And here again you can see that this at the 5' of this new nucleotide and the 3' of this already present nucleotide, a bond will be formed that is called phosphodiester bond. The remaining pyrophosphate ion, they will be removed and they will be converted into the phosphate ion. So this is elongation. Once the nucleotides they are continuously being added to the growing DNA strand, this process will be called as elongation. And here you can see that core enzyme which are A', E' and Q, they are basically catalyzing the synthesis of leading and lagging strand. The nature of the chain elongation is, it is basically the formation of phosphodiester bond, series formation. Here this is one phosphodiester bond, this is second, this is third. So this is basically the series of phosphodiester bond that will result in the formation of elongated DNA strand. Here you can again see the leading strand DNA synthesis, and here is the lagging strand DNA synthesis. Here you can see that DNA is synthesized in the form of the short fragments, and those short fragments, they will be later on joined by the DNA ligase enzyme, and hence we will have an intact form of the DNA strand. So this is the difference between the leading strand DNA synthesis and lagging strand. If we look at the lagging strand synthesis, RNA primers, they are forming the Okazaki fragments, and later on those are digested by RNAs, and gaps are filled by the DNA polymerase 1. So it means that DNA polymerase 1 is also performing the ligase activity as well. It will move from 5' to 3' direction, and it will seal the gap or the any type of the gaps that are actually left behind as a result of lagging strand synthesis. So the nick between the 5' end of the one fragment and 3' end of the next fragment is sealed by the ligase enzyme. So that's how the ultimate gap is filled by the ligase enzyme. If you look at the Okazaki fragments, these Okazaki fragments, they are synthesized on the DNA template strand having the 5' end, and these DNA fragments, they are called Okazaki fragments. Here you can see that they are synthesized in the form of the small fragments, short fragments. These blue arrowhead, they are actually showing the Okazaki fragments. So they are synthesized from the 5' end of the DNA, and they are synthesized in the form of the short fragments, and those short fragments are called Okazaki fragments. Under 1000 to 2000 nucleotide long in prokaryotes, and 100 to 150 nucleotide long in eukaryotes. So their length is larger in prokaryotes as compared to the eukaryotes, where they are consisting of 1000 to 2000 nucleotide, whereas in case of eukaryotes, their total length is ranging from 100 to 150 nucleotide. The daughter strand, which is consisting of Okazaki fragments, that will be called as lagging strand. So here, this is lagging strand, and this continuous strand, which is being synthesized from 5' to 3' end, it will be called as leading strand. So this is the difference. Here we can see another structure, another diagram. Here you can see that DNA topoisomerase 2, which is also called as DNA gyrase. It will remove the supercoiling. When the DNA is basically unfold from here, it will result in the formation of supercoil in somewhere here. So here you can see this is the DNA B, which is helicase enzyme. It has bounded here, and it has separated the two strands from each other. Once it has separated the two strands, now the single-stranded binding protein, they have bonded to this single-stranded DNA. In the next step, RNA primer will be there, and this is primase enzyme, which is synthesizing the RNA primer. So after the synthesis of all these requirement, here you can see on the lagging strand, this RNA primer is forming the short fragments, Okazaki fragments, whereas on the leading strand, a continuous structure is formed. So here you can see that lagging strand synthesis, it will in the form of these small fragments, which are called Okazaki fragments, whereas in case of leading strand, we will have a continuous synthesis of the DNA. So the last step of the replication process is the termination of the replication. Once the termination sequences, they are reached at the double-stranded DNA, it will result in the termination of the replication. So replication of E. coli, as all of you know, that it is basically in most of the cases, Whether we can see that it is in most of the cases replication is bidirectional, mean it will start in both direction. Here you can see that in this case it is replication will be started from this direction to this direction, and this in this case it will be from this direction to this direction. So in all of the cases the replication is bidirectional, and here you can see in the E. coli that this is the origin of replication. This is one direction, this is counterclockwise direction, and this is other direction which will be called as clockwise direction. Now this is counterclockwise fork-like structure or fork-like form, and this is the clockwise fork tape of the structure. So if the two replication fork, they will meet, they must meet at a point. These are the two replication fork, and they are basically they will meet at this specific point. This is the counterclockwise replication fork, mean this is moving in this direction, and this is the clockwise replication fork. So these two will meet each other at one point, and this is TR at 32. You may call this position here, this position where these 2 points they are meeting with each other. All the primers will be removed, and all the fragments will be connected by DNA polymerase 1 and ligase. So later on, once the initiation has been completed and the replication process that has entered into elongation phase, and a huge number of the nucleotides they have been added to the growing polypeptide chain, and ultimately the termination sequence will be reached there. So at the termination sequences there are TER binding proteins, specific type of the proteins, those are called termination site binding proteins. They will recognize the termination sequences, and ultimately the process of termination will be achieved with the help of these termination sequences. So that's all about the process of replication in case of prokaryotes. If you have any question, you can ask, please.
सर यहाँ पे आपने बताया था कि जो डीएनए जो होता है वो रिकॉग्नाइज करता है और ओआरआई साइट को सी और उसके बाद डीएनए बी और सी बाइंड करते हैं वहाँ पे, और लोकल 80 रीजन जो रिच रीजन होता है वहाँ पे बाइंड करते हैं.
यस.
तो जीसी पे बाइंड नहीं करते हमेशा, 80 पे बाइंड करते हैं.
नहीं, 80 रिच इन द सेंस कि 80 वहाँ पे ज़्यादा होगा, इसका हरगिज़ मतलब ये नहीं है कि जीसी बिल्कुल मौजूद ही नहीं होंगे. जीसी कंटेंट विल ऑलवेज बी देयर. यहाँ पे अगर आप देखें ये आपको सी भी नज़र आ रहे हैं, ठीक है ना, ये आपको सी कंटेंट, ये जी भी नज़र आ रहे हैं, बट मेजरली एडेनिन एंड थायमिन आर देयर एज कंपेयर टू द ग्वानिन एंड साइटोसिन सीक्वेंसेस. ठीक है, तो इसी वजह से हम इनको 80 रिच रीजन कहते हैं. बट 80 रिच रीजन डज नॉट मीन कि उसमें जी और सी कंटेंट बिल्कुल मौजूद ही नहीं है. जी एंड सी आर आल्सो देयर, बट मोस्ट कंसंट्रेटेड न्यूक्लियोटाइड्स आर एडेनिन एंड थायमिन. क्लियर है?
यस सर.
Okay, any other question? If you have any other question, you can ask, please. No question? Class. Okay, एक तो डिसाइड करें कि आपने क्विज कब देना है. असाइनमेंट की तो डेट मैंने आपको दे दी है.
सर ईद की कितनी छुट्टियां होंगी?
अगस्त 3. ये थर्सडे से स्टार्ट होंगी. यू विल हैव हॉलीडेज ऑन थर्सडे, फ्राइडे, सैटरडे. नहीं, थर्सडे को नहीं, फ्राइडे, सैटरडे एंड संडे. From Monday, your academic activities will resume. दस तारीख ठीक है. 10 अगस्त को हम ले लेते हैं. ठीक है.
ठीक है. ओके सर.
10 तारीख को फिर क्विज सब लोग जो है ना उसकी तैयारी कर लें, ठीक है। और अभी कुछ और भी लेक्चर्स इन बिटवीन होंगे आपके, तो उनको भी हम जो है वो शामिल करेंगे।